the different IBRO regional committees. So I will start this morning by introducing our first speaker, who is Dr. Simona Lodato. She is actually an associate professor and group leader at Humanitas University and Research Center in Italy. Simona's research focuses on cortical diversity and neurodevelopment disorders using advanced 3D human organoids model and single-cell omics approaches. She was awarded an ERC starting grant in 2022, and she investigates how cortical diversity affects early cortical activity. She is a proud alumna of the Fence-Calvi Network of Excellence and serves as the Next Generation Leader Advisory Council at the Allen Institute. She's also an advocate for diversity in brain science as an Alba Network ambassador. Join me this morning in welcoming Simona to give her talk. And Simona, please take it away.
Thank you so much, Olamide, for the very kind introduction. And I'll share my slide. Share my screen. And I wish to thank you and all the organizers for IBRO and the ECC for having me here today. I'm really pleased to be joining you at whatever time of the day, evening and afternoon this is, to tell a bit about our work on human organoids. So as Olamide already introduced, I've been long interested in the cerebral cortex, which is the part of the brain that has undergone the most pronounced expansion during evolution and has solved many of the different functions that make us humans. We have known for over 100 years, however, that the cerebral cortex is populated by neuronal and non-neuronal population, and among the neurons, it was quite easy to appreciate already the morphological level from Ramón y Cajal, another Spanish neuroanatomist, already over 100 years ago, that we had different classes of cells, of neurons, that were largely distinguishing excitatory projection neuron, very long projecting, and inhibitory interneurons. And we know now that the interaction between these two types of large classes of neurons that populate the different layers of the cortex is critical for maintaining a balance between excitation and inhibition. And indeed, interneurons, despite being much smaller in frequency, they modulate the activity of the excitatory neurons. An alteration of this balance in any direction has been associated with the onset of neurodevelopmental disorder, neurological conditions like epilepsy, as well as late onset disorder as schizophrenia or ALS, or even psychiatric disorder as depression. But we also have recently learned with the advent of molecular neurobiology and specifically the single-cell omic era, that this is just the tip of the iceberg. Indeed, the degree of diversity at the neuronal level that we have in the cerebral cortex is much, much larger. And here I'm far from explaining it for you today, but just I would like to highlight that despite an intensive work of the entire community, we do not have a unifying criteria to define a single neuronal subtype, but we know that different neuronal types populate the cortex, and their diversity stems for the integration of many different features, their morphological features, their physiological properties, as well as their molecular properties that we now can appreciate with single cell resolution better than ever before, and with different type of modalities, transcriptomic, epigenomics, and we're getting to the proteomics too. And this is very important because if we think about the central nervous system, in particular the cerebral cortex, all this diversity is generated during a very limited time of development, very early on, from a limited pool of progenitors. So this entire diversity stems from a relatively small group of cells, and eventually is determined to create these circuits that modulate the excitatory inhibitory balance. And that's exactly what my lab is interested in understanding, how this complexity arises and what goes wrong in developmental, neurological, neuropsychiatric disorder. And in particular, it has been established that there is a strong genetic component, so master regulator, transcription factors, and even, epistatic relationship between different type of transcription factor can control the identity of individual neurons, which eventually serve distinct circuits. And also activity-dependent processes as well as environmental cues can help shaping these circuits and this diversity. And more recently, we've become interested in understanding a specific type of activity, which is namely the spontaneous activity, which is a prominent feature of the immature brain. As these neurons, these cells that are so special for us and so diverse, they start firing very early on, even before the circuits are actually established in the cortex. And while originally was assumed that this early firing was just sort of a treadmill for the neurons to start exercising, we now know they might have profound consequences on the definition of these circuits. So since by training I'm a molecular biologist, we try to address this question. We wanted to address the question of what are the substrate for this early spontaneous activity, which is known to be present in the immature brain, but it doesn't really, we don't really know where it stems from, how it's articulated, how this activity can help establishing this circuit. So with a brilliant postdoc in my lab, Sara Mancinelli, we decided to take a specific angle to this question and start profiling a single cell resolution, the cerebral cortex for many different stages of development, both in mouse and as well as in fetal. And what you're seeing here, the different cluster in different colors here of different cells that we know emerge from early in development, and we can find them also later during the differentiation of these cells. And I just want to tell you that from these analyses that involve different hundreds of thousands of cells, Sara managed to not only depict how the molecular divergency, the diversity of these cells emerges, but also identify new factors that can play in the diversity. And among those, we found ion channels. So I'm not going to tell you much about these because most of the work is done in mouse, and I'd like to show you more about our work on organoids. But the idea here is that we found that ion channel is a category for long overlooked, as actually very important in defining the identity of different classes of neuron, even in neurons that are still developing. And among different kind of channels, we focus on one specific, which is HCN1, which is a channel that is expressed by a specific population very selectively, here in mouse at least, and start looking into it. And the reason why we start looking into ion channel and more specific into this one is because ion channel role during development remain largely unappreciated and unknown. We tend to study it as a community in the adult circuits where many things are actually different, and also that ion channel are among the most divergent gene families in terms of expression that differ between mouse and humans. A more intriguing for us on a translational aspect is that these ion channels, including HCN1, are actually targeted or specific mutations that are de novo most of the time, and they are associated with diseases, developmental epileptic encephalopathy condition, which are characterized by recurrent and unprovoked seizures, usually untreatable with any drugs, associated with many comorbidities, including neurodevelopmental condition, motor delays, intellectual disabilities. And I always like to highlight that originally these type of disorders were named just epileptic encephalopathies, and it was the clinical side that really pushes for a change in the nomenclature of these diseases, acknowledging that there was a developmental component that was largely unappreciated and needed investigation. So you can, I hope I convince you by now that we really wanted to move into some type of modeling for investigating this disorder, and as well the function of these genes in humans. So let me tell you what we know about this rare condition. And this is relative to HCN1, but there are many different types of ion channel mutated that present the similar circumstances. Despite the fact that the gene has been identified less than 10 years ago to be causative of this disorder, we have already found more than 80 de novo variants. There's no genotype-phenotype correlation, and it can stem as early as birth, and sometimes even mothers complain about feeling the seizure in the womb already. A very poor outcome most of the time. In certain type of mutation, we have neonatal death and a severe neurological impairment. And in some cases, we even find association with structural lesion, meaning we can see aberrancy in the morphological features of the brain, such microcephaly. And as I told you, no treatment, because most of the time are not only pharmacoresistant, but these patients develop a sort of paradoxical effect with the usual anti-epileptic drugs. These all motivated us to generate urgently a model for developmental and epileptic encephalopathy. And that was the moment when my lab turned into organoids. And as a developmental biologist, we look at that on sort of a different perspective compared from cellular biologists, because we really wanted something that could model faithfully not just the brain, but the development of the brain and the different component of it. And aware of the kind of diversity and complexity the brain has, we were really looking for something that was showing the degree of heterogeneity that we had. So this was a few years back, and we had a choice of few organoids that were already organized. Those are three-dimensional structure, where basically we start from pluripotent stem cells, either ES, embryonic stem cells, or induced pluripotent stem cells, and you can fate them to organize themselves in a very sort of unpatterned way without providing any type of information, and where the complexity might be higher, but the consistency of your protocol is actually rather limited, or you can have some degree of diversity just within certain regions. That's the choice we went for, and we call them pattern organoids. The key of this kind of experiment in any of these protocols is that you're not only relying on external cues that you can provide to your culture, but these cells sense each other and inform each other from on what they will actually become. And within a certain lineage, the cells can acquire many different types of fate. So that's what we decided to do. And here I'm showing some of the preparation we have along this protocol, where we have steps for induction and differentiation and maturation. And despite the fact we do pattern our organoids toward a cortical identity, so more frontal and dorsal identity, we basically, after just the induction time, we let the cells develop in a sort of a very naive and uninstructive way. And this work is the work of a very I think resilient and hardworking student in my lab, Elena Florio, who has been generated for the first time a bank of iPS derived from patients and isogenic clients and parents, relatives for these kids that carry the HCN1 mutation. So here's what we do. We culture them for several months, and the idea is to characterize the different organoids and try to model the disease in vivo, but also the developmental aspect. And the reason we chose this organoid, which was originally developed in Paola Arlotta's lab, which was my mentor when I was at Harvard University, is because they actually faithfully recapitulate not only different steps of corticogenesis, so we find the presence of cortical neurons, cortical progenitors, even human-specific progenitors, but they also display the presence of non-neuronal population like astrocytes of the neural lineage, which we can see emerging at specific time in the culture that is following human corticogenesis also in temporal step. In addition to that, we can see the emergence of different classes of neurons. I'm not going to spend too much time on that, but as I showed you at the beginning, we do have markers to discriminate the presence of different populations and neurons that we can fully characterize in the fetal cortex, as well as for those that are conserved in the mouse cortex. And we see the emergence and a primordial lamination of these neuronal populations also retain in the cortex, in this cortical plate of the organoids. And lastly, which was very relevant for our question, these organoids display spontaneous activity. I hope you can see here, those are calcium cells that are labeled with a calcium sensor, which can appreciate the differences in calcium dynamics. And you can see here, we can see neurons activating and displaying different variation of calcium as a proxy of their activity. So this was critical for us since we wanted to recapitulate aberrant activity of these patients. So Elena differentiated the first derived, the IPS, the induced pluripotent stem cell, from different patients. And the moment we had 8 different lines. I'm going to show you here some of the results. And then she generated cortical organoids for all of them. And of course, for all of them, you have to have multiple batches to really understand the consistency of your protocol. I hope you can appreciate here in our control condition, we have these beautiful rosettes, which are the organization of the human cortical progenitors. And then we have neurons that are labeled here with maps. And you can already see by this immuno that in the 2 mutations I'm showing you here, which are distinct mutations from different patients, we do not see the same kind of nice arrangement of the rosettes and also this organization of the neurons. This is now under investigation in terms of quantification, but also we have single cell analysis to appreciate what is the overall composition of these cells. But it's already very promising to show, to confirm that among those patients, those that had microcephalic fissures, so smaller brain, are recapitulated in terms of growth rate in the organoids. So we could see a lower curves of growth over 4 or 5 months of development in the lines that were corresponding exactly to this patient, and not in those that were actually not showing any alteration. And when Elena starts analyzing the activity of these organoids, we had to wait up to 4 months because of the neurons have to mature and the astrocytes have to start growing into the organoids. But I hope you can see here, we have these waves of activity. Again, we are looking at a calcium sensor here, and then we can quantify the frequency and the amplitude and the mean distance of the firing activity. And this in the mutant, you can see that it's very different. It's much more frequent. The flickering is actually distinct, and you can even appreciate it in live. The interesting aspect is that we didn't see the same pattern of activity in all the different mutations, but according where the mutation is falling in the channel, we can see different type of profile and different type of alterations, which makes these organoids an important platform even for personalized drug screening. And I hope I convinced you by now that the cortical organoids can be generated and they're critical for modulating development and alterations in different patients. In our case, it's a developmental epileptic encephalopathy, but this can be done in many different type of mutation. We have seen distinct effect for those mutations that were present in the pore membrane rather than in the N-terminal of the domain. And there are alignments in the expectation of the growth abnormality compared to the structural defect that we found in the patient. And given the role that we are also investigated in vivo, controlling fundamental aspects of neuronal diversity, we are now leveraging on single-cell transcriptomics to understand how the cells are generated also in the organoids and the different mutations, and try to see how the circuit assembly and the differentiation program are altered. But I'd like to leave you with an idea of the power of these models, which we are clearly exploiting in this case, but also in many other directions, and for many different conditions, including the gene editing. You can alter. In some of our cases, we did gene edit to correct the mutation to obtain our isoform. Isogenic lines, but we also study neuroconnectivity and function. You can study brain tumor in these organoids as well, and you of course can study at the molecular level, as I told you earlier. But of course, those models are not perfect, are very useful, and they still present a lot of limitations. They first have the limitations that they only contain spontaneously the neural lineage. So everything that is originating from the neural tube, meaning progenitor cells, neurons, astrocytes, and in some cases oligodendrocytes. However, they do not contain other population, other cells that are still present and very relevant in many of these conditions that we explained, but they are not deriving from the same embryonic layer. And I'm more referring to microglia, for example, or blood vessels. So there are many strategies, complementary strategies to include cellular diversity. And I'd just like to give you a couple of examples that we're undergoing, for example, for vascularizing this organoid, which is critical, having the endothelium presence and eventually vascularization in long-term culturing. It's fundamental for maintaining and the activity and maturation of these neurons. We have first established a protocol to insert a UVEC, which are endothelial cell line vascular to promote vascularization. I hope you can appreciate here that in these vascularized organoids, we managed to form some circular structure, a tubular structure like vessels. But then we scale it up to iPS-derived endothelial cells that we can fuse together, sort of merge in the hope of generating blood-brain barrier. And here you can see our cortical organoids with the specific rosettes that we can characterize also molecularly here, and then our vascular organoid that is growing and stemming this nice vessel, and eventually when you merge them, you can appreciate this CD31 positive staining for endothelial cells within our assembloids for endothelial cells. And much more needs to be done, but I wanted to give a hint of how this can happen. And we're also pairing up with engineers, and there is a huge interest, I have to say, from the engineering part to try to complex this strategy, these models, and achieve strategy to increase diversity and more precise architecture, leveraging bioprinting technologies. These are, many different types of bioprinting techniques and even devices that can leverage on sacrificial networks or just simply printing multiple populations at the same time. And the example I'm showing you here is, again, our organoid-derived cells and some endothelial cells seeded in a specific geometry. And we can keep them in culture for 4 months, and they would continue surviving and integrating and remaining active from functional level. So I'm sure much more needs to be done, and these are not to be called mini brain or anything like that. They're just a simplistic model to appreciate the complexity and diversity of the central nervous system, and in particular of the cortex in our case, that offer an invaluable opportunity to sort of open a window into an organ that for the longest time has been completely precluded to us, which is the developing human brain. And with this, I'd like to conclude. I already mentioned a few people whose work I've highlighted here, but most of it I'd like to thank again IBRO and ECC today. And I've had the opportunity to collaborate with IBRO already in the past at our Fans Forum 2022 together with Shrikant here. It was a wonderful experience, and discussing about the colors of the rainbow and how diversity matters. As you can see, it's a topic that is very close to my heart, also in my science. And I'll be happy to continue collaborating with IBRO and ECC for the future. With that, I hope I'm on time, and I'm happy to take any question.
Thank you, Simona. That's a great talk. We will leave the discussion and Q&A at the end. So, okay, yeah. So now I'm going to introduce our second speaker for today. Our second speaker is Dr. Thomas Tagoe. Dr. Tagoe is a neuroscientist and science communicator based at the University of Ghana, where his lab is focused on elucidating mechanisms underlying experience-dependent plasticity in health and disease. Is among the first cohort to be awarded as part of the Welcome IBRO Neuroscience Capacity Accelerator for Mental Health program. Through this award, he's working with his collaborators to set up the first electrophysiology lab in Ghana. Thomas is the regional president of the Ghana Science Association and outgoing member of the IBRO Africa Regional Committee. So please welcome Thomas. Thomas, I'll leave the floor to you.
All right, that's great. Thank you very much, Jack. So I'll hand it over. Okay. Okay, all right, that's great. Thank you everyone, and thank you to the ECC once again for this opportunity. Just to share some of the work that we're doing here out of Ghana, specifically in the area of just trying to look at non-pharmacological therapeutic approaches to treating depression. So for this talk, what I'd like to do is just briefly just touch us back on the need for why it is we should even be exploring alternative therapies, and then share some of the data we're currently working on in the lab to look at how we are investigating these alternative therapies and the evidence and support for it. And lastly, look at the direction that we're really taking now, which has been a bit supercharged thanks to the IBRO support that the lab has recently received. So it's just to take us back a bit, it goes without saying that, depression has really received quite a lot of attention over recent years, along with other mental health ailments. We're expecting that by 2030 is going to be one of the leading causes of mortality. So there's quite a lot of work and funding going towards trying to really understand depression and treatments. Now, one of the things is that there are quite multiple pathways that, lead, for example, from stress to depression and anxiety. And beyond just the nervous system dysfunction that's common, there's also, other systems that also experience some form of dysfunction, whether it's the digestive or the endocrine systems. In people with depression, it does seem to be a problem. So there are currently available therapies, which is well known, but it's also well known that a lot of side effects come along with it. And then you also have a small percentage, somewhere like 10 to 15% of people who may be unresponsive to a lot of the available therapies. So all of that makes a case for, trying to explore not only alternative therapies, but then also non-pharmacological therapies, because at least the nervous system is very unique in the way that both substances and non-substance experiences can then modulate its function. So one of the first areas that we started off from was to try and explore the gut-brain axis. Okay, so it's now well established that microbes, for example, are able to mediate and inform the relationship between the gut and the brain, so that changes in gut microbiota, for example, can influence mood disorders, and that's an area that's really been explored to try and best take advantage of this knowledge that we keep on uncovering every day. So because we wanted to explore probiotics as one potential avenue, we started off with a question of the various bacterial strains that have been shown to have benefits, which combination could we use and what concentrations could also be most effective. So the model of study that we use, the mouse model, specifically we use the chronic unpredictable mouse stress model to induce depression. And this is what a typical protocol looks like. We will run it for about 6 weeks and try and change the stressors on each day, which is then randomized to prevent anticipation from the animal point of view. So you have a mixture of these different things, right? Something like food deprivation, water deprivation, and so on. And then over that 6-week period, we have a 2-week period where it's just purely the stress. And then the 4-week period after that is then the stress plus whatever intervention that we put in place. And at the end of the study, we collect a number of different measures to then be able to assess how effective was their therapeutic intervention. So for the probiotics, as an example, we looked at a number of different things. First, we looked at weight over the 6-week period, and this looks like a lot of different groups, but what we had was control groups here in blue, stress group, fluoxetine, which is a standard antidepressant that is used. It's a 5-HT reuptake blocker. Then one strain of bacteria, which is the lactobacillus, a low concentration of it and then a high concentration. And then another strain, which is a bifidobacterium, low and a high concentration. And then lastly, we mix up these two strains and then either a low concentration or a high concentration mix of it. And we can see that the weights for all the groups that were stressed do pick up at the end, except the stress-only group. Now the classic test for depression is a sucrose preference test to test for anhedonia. And once again, you can see that yes, the stress group were given quite low ratios, but then fluoxetine and then high concentrations of either the monobacterial therapy or the combined therapy increase the sucrose preference. We continued on to then do a number of different tests, which we used to, of course, assess the extent of depression and associated anxiety. So, for example, the tail suspension test, we see that indeed the high concentrations were able to reduce the mobility time, similar to the fluoxetine in the control group. Also, open field tests, where the time spent within the central zones, we can see that this same fluoxetine, the high concentrations. And interestingly, of course, you start noticing that the low concentrations mix of the two bacteria sometimes seems to perform equally as high as the high concentration. We also did a hot plate test for sensitivity, and we see that the probiotics intervene with that, and also did a measure for BDNF levels in the prefrontal cortex. And it didn't seem to really be any difference between all the groups. We had just like a small N numbers for this. So we're interested in going back and increasing the power of this to see if it really stands true. So having established this, we then decided, okay, so this is going to be the probiotic concentration we're going to use. We're going to use a high mixed concentration because that seems to give the most consistent effect across all these parameters we were measuring. Now, the next intervention, of course, which is much more interesting in our lab that we study, is environmental enrichment, where environmental enrichment refers to this idea of enhancing just the living conditions of the animals that we're working with to increase, sensory, social, cognitive, and motor stimulation. And a lot of data seems to suggest that these sort of interventions increase neurogenesis, modulate plasticity, but also modify the HPA axis response to stress. Now, there are a number of different protocols existing out there for environmental enrichment. It's quite an array and there's very little standardization within the field. But one of the challenges that has made it difficult to translate is, putting the animals in an enriched environment for prolonged periods of time, meaning clinically it becomes a bit tricky to do the same with patients or clients. So we wanted to see if something more practical, like 2 hours daily enrichment, could offer some of these equal benefits as the long periods have shown. Okay, so the enrichment that we were doing, you have these sort of like cages where it's a slightly larger cage than the small ones the animals are kept in. We started off with just one running wheel, and we realized that they like to hog the running wheel sometimes and not share. So we decided we must make it two to encourage some sharing and good behavior among the animals. But of course, put a number of different things in their mazes they can explore inside, some climbing ladders, piping, and so on. Okay, so now what we then did was now we're working with a smaller number of groups, right? We have just a control group, stress-only group, those receiving the probiotics, which was that high mixture, the enrichment group, and then fluoxetine also receiving. And this time we're collecting data at multiple time points again at the start before we begin the study, at the 2-week mark following the stress, and then midway through the intervention period and at the end of the intervention period. And the measures we're taking similar as before, right? Weight, sucrose preference test, doing a number of tail suspension, open field, novel object recognition, Morris Water maze, and then also some neuronal volume measures. Okay, And here's what we seem to be getting so far. So firstly, just in terms of the weight, so both probiotics in purple here and enrichments in orange, both of them seem to be equally preventing the weight loss even after 6 weeks of this chronic unpredictable stress. And you see at the end of the 6 week time point is really only the stress-only group that has significantly lower weights. Okay. Next is anhedonia, so the sucrose preference test. We're seeing that it's equally reversed whether using probiotics or enrichment. So once again, our stress group was quite low, but at the end of the period, we find that equally, like the Fulton, probiotics or enrichments are great. And you see that here with a 6 week time point that the stress only group is resulting in quite significantly less ratios, which is evidence for anhedonia. Moving on from that, this is from the tail suspension test that we did. So immobility time, the stress only group has significantly higher immobility time. Here's where we start noticing potential differences between the probiotics and the enrichment. Because whereas the curling time seems to be the same across board, the swinging time appears that the probiotics swinging time is much less. Now, this matters because swinging behavior seems to be mediated between something like your dopaminergic system and your serotonergic system and so on. Whereas curling behavior is more mediated between like your GABAergic system inclusive. So this alone seems to suggest that, okay, so although your probiotics and your enrichment may, on the surface, be equally sort of like preventing anhedonia, as an example, they may be doing it through slightly different means. Okay. Want to then look at memory deficits because short-term memory deficits are something that's recorded in depression. And using the novel object recognition task, yes, the stress group had lower discrimination index, but it's really the enrichment group which seems to have the highest discrimination index. And then with the Morris-Water maze, it seems pretty much equal where both the probiotics and enrichment are reducing the time that it was taking these animals to locate the platform on the last day of training. So this on day 4 of training. So this seems once again to be leaning towards the idea that the probiotics and enrichment may be doing slightly different things. The end goal is the same, but the path leading to it may be slightly different. Okay. We did the Y maze test as well, and it doesn't seem to have anything clearly standing out. Seems to be a bit of a trend where it may appear that the enrichment may be, for example, encouraging more exploratory and reinforcing that short-term memory, especially with this time spent in the previously closed arm. But these differences were not significant yet. We're looking to increase the power of it to see if we are missing something here. Okay. Then the last thing we then also did was this measure, right, for cell density. This is just showing the data from the prefrontal cortex. We're currently analyzing some additional work we did with the hippocampus to see if this neuronal cell counts and density is the same or not. But what you will notice here is that once again, it appears that the enrichment is given as a higher cell counts in the prefrontal cortex. Now, looking at this, what we really started thinking about was the fact that even the control conditions, which were not stress, they clearly have less cell counts in the prefrontal cortex as compared to the enrichment. Now, this often was a bit strange because you'd think that it should be, the enrichment should be protective, but not so much positively different from the controls. And it made us start questioning, the control conditions that animals are normally housed in, right? There's sufficient evidence out there suggesting that, keeping the animals in smaller spaces, in lower numbers in the cage, that in of itself, what we've come to accept as standard housing conditions, may even be a stressful housing condition. Because it's very different from large space where a lot of enrichment in this space. So it brings into question the need for us to really start questioning what do we consider as standard housing, and is that in of itself dysfunctional in some regard. Okay. So in the last minute or so, let's just wrap up with what we have, which is right at the start, we've shown that it appears that your probiotics are very much a dose-dependent effect. Not only that, but combination therapy could be good to reduce the dosing that's required. Next to that, it appears that 2 hours enrichment is enough to induce these beneficial effects. So now we can start exploring it more in terms of what are the mechanisms mediating it, and how can that really be translated clinically. But also, if it appears that the probiotics and enrichment may have different mechanisms, so we then explore this relationship. Okay. And by exploring it, here's what I mean. Could it be a synergistic effect that the two could work together so that by using environmental enrichment and some combination probiotics, you'd be able to, have a therapy specifically personalized for people. But beyond that, we really want to refine the enrichment protocols, like I said, going to the campus, see what's happening over there. And more importantly, really start exploring electrophysiological properties and changes. As was mentioned right at the start of the introduction, like Jack said, we recently had an ePhys rig set up in the lab here in Ghana, and it's the first of its kind. So that's myself and Joseph Raimondo from Cape Town UCT. This was very recent. We got this rig set up, all facilitated by the IBRO Welcome Grant, so that with this, we can now start doing some electrophysiological recordings, find out how is the enrichment affecting neuronal cell properties, which is then mediating some of this learning and memory changes that we're seeing. But then also, what is the probiotics doing differently? So we can start identifying, is it potentially synergistic in nature? But beyond that, there's a need to also then start considering how to translate this clinically. Can we design personalized enrichment experiences for people, which can then also be used to inform therapy? So I'd like to just end off by just acknowledging the guys in the lab. This is very hard at work, of course. And just the team behind it, Leo, Emmanuel, Andrew, there are a few people missing here because the lab has just continued to grow since the time that this data was taken. So thank you very much. And yeah, we'll just stop it here and wait to take some questions from you all. Thank you.
So, okay, thank you very much, Thomas and Simona, for these wonderful talks. We have lots of questions. I will try to filter a little bit and maybe to group the questions. And the first question is to Simona. And when comparing gene expression profiles, how similar are the neurons in organoids to the normal human neurons and to those in patient populations? And how do these compare in the usability to the live animal models?
Thank you so much, Manuela. I think it's a great question. People have done it for different protocols, as was stated there. And it seems to be quite consistent that at the transcriptional level, even epigenetic level, we can reach a fairly comparison in alignment with different kind of populations with fetal cortex, and in some cases, long culture, even just after birth. We don't have mature brain. In terms of the disease condition, this has been done in certain cases, and despite the fact that we don't match organoids with adult cortex, you can identify whether disease traits that are confirmed, whenever the disease is available. And this is most of the cases neurodegeneration, where we have material from patients that can be assessed. And the comparison with models, I think people are investigating right now how the mouse organoids are actually recapitulating the stages of development in mouse, and things may be different because mouse, yes, are slightly more complex to culture to a certain extent. But definitely, it's again, we have models, we don't have the entire brain, and I think we're never going to have, and I think it's important to state also for ethical concern. But it's a good cellular model when we can study at least up to circuit level some condition. I'm trying to be fast so you can ask more questions, but let me fetch them more. Yeah.
Thank you very much. The next one for Dr. Tagoe. Can you explain a little bit about the use of male or female mice, and what about the age? If your results are obtained with younger animals, and what do you think would happen if they use older animals?
That's a great question. So this work was done with, I guess, what you would call adolescent mice, because they're around that 6 weeks. Well, we started when they were around 6 weeks old, and we're all male, unfortunately. Sorry, people. But yes, there's that need, of course, for us to do some of this work with females, because there's enough evidence that suggests that it will be different between male and female species. With the adults, I will expect that maybe the enrichment programs will have slightly less effect in the adults, only because we know that all the mechanisms and the life plasticity seem to get less strong or less prominent with age. But I wouldn't discount it completely, because at least in human studies, it has been shown that some of these environmental enrichment components, for example, the increasing physical activity, specifically, there's some evidence for that to reduce evidence of depression in human adults. So it's just a matter of fine-tuning and refining it so that there's more consistent evidence and results. Yeah.
Okay, thank you very much. Simona, there are several questions here about the protocol, so I'm trying to summarize. There are different protocols available for cerebral organoids. What types of protocol do you use? And what else here?
I think there's a question for IPCs as well, IPS.
Yes, and
So
the question
I'll start.
the type of cells, and yes.
Yeah, I'll start on the IPS. So we did get them from patients, meaning we started from blood samples. When you deal with a neurodevelopmental condition, a neurological disorder that impair kids, there is clear resistance from parents, rightfully so, to perform any biopsy and extra procedure on the kids. So we try to work with a very limited amount of blood, and it's sufficient to have basically 2 ml of blood to start from monocytes, circulating monocytes, the differentiation of IPC. So we normally use the Sendai protocol, which is basically a kit that originated from the stems from the original factors, Yamanaka factor, that were present in the Nobel Prize award, and it's quite consistent. It takes a bit though, and it's pretty time consuming. So you have to come and change media daily, including Saturday and Sunday, which is something that doesn't make students very happy. But you can start virtually from any cell, actually, any cell of the body. Probably you start from a neuron, which usually is not the case. It would be the least effective, but any other differentiated cells will work. And then you have to pass these cells for a while, and you have to make some QC, which I strongly recommend to do. And I'm happy to answer question on that, offline as well. I left my email there if people need to know. I think the good point is that we have no evidence that to study neurodegeneration, I think there was a question about, if I'm studying neurodegeneration, do I have to start from an older patient? I think we have no evidence that this is actually a problem, because the reprogramming process is somehow deleting the epigenetic landmark, hallmarks, and you will sort of scratching everything from zero. You're starting again from zero. So this shouldn't be an issue. There might be some memory in the cells, but at the moment doesn't seem to interfere with the differentiation. And in terms of protocol, it's true we're using the cortical organoids, the sort of Wnt inhibition and TGF-beta inhibition protocol. We are trying to basically push toward the cortical identity, and this protocol relies on the signaling that we've studied for many years in the neural tube to define different regions. And again, this is also published, so I can definitely send it around.
Okay, thank you. Another question for Dr. Tagoe. Why do you choose to study the frontal cortex, and if you examine any other brain areas? And what about, what's the other one? Sorry. did you check anything about myelinization in your experiments, or are you planning to?
Yeah, so really we wanted to look at the hippocampus and the prefrontal. The prefrontal data was just ready first, not the hippocampus. So yes, when we do, this hasn't been published yet. When it will be, we'll include the hippocampus data as well. The understanding being that a lot of the learning and memory being hippocampus based, and then like your decision making and so on being prefrontal cortex area. So we were going to look at both of them. And then also look at the two sexes. And in terms of what was it, myelin? No, we haven't considered that. But like I said, we now have at least the capacity within the lab to not only do the electrophys aspect of it, but also some very decent images so that we can start looking at some additional markets to see if there are any changes in that regard. So yes, that will be considered going forward.
Okay, thank you. I think that we have time for one more or maybe two more. Let's Simona, what about the relationship between the mutation you studied and the structural disruption? I don't know, you said that there is no relationship between the phenotype, genotype, but do you guys know what's the functional effect of these mutations?
No, that's a very interesting question. We are trying to understand what could potentially go wrong. First of all, in the localization of the protein, of the channel on the membrane. So we're studying this also in heterologous system as well as in organoids. But one potential uncovered role for this channel is also to be involved in cell cycle and to modulate, these sort of early activities also present at the progenitor levels. At the moment, for HCM1, it doesn't seem to be expressed, at least at a high level in progenitors, in precursor. We definitely find it in neurons. But we are investigating if this could be a non-cell autonomous effect, or rather an effect that can be observed later on. And we do see effects on the morphology of the neurons, and probably on the organoids. The expansion of the arborization of the neurons might also reflect how these cells will eventually grow later on. But I don't have, for now we all have hypotheses, not real data yet.
Thank you. And I think the last one for Thomas. Do you think that probiotics and the enrichment environment can help in Alzheimer's disease patients?
I'll say why not. I'll say specifically with the enrichment, because it's being considered in multiple different neurodegenerative conditions and neurodysfunction. I'm not sure to what extent it will go to, but I think we just go by the foundational principles that the enrichment is enhancing all these neurogenesis, plasticity, activity, and so on. Then it makes sense that it could offer some benefits, maybe not as a therapy on its own, but possibly as an adjunct to any of the new stuff that's being developed to sort of make them more effective. But I would say it's definitely worth exploring.
Okay, so thank you very much again.
Thank you. So if there's questions that cannot be answered, if you have more questions, please feel free to contact Simona and Thomas. I'm representing the IBRO ECC to thank the two speakers for their wonderful talks, their wonderful projects, and I also want to thank the attendees today. It's very good discussion, and we have very good attendance. I think it's a very lively discussion, and we get attendees from different parts of the world. So this is very exciting. And I also want to thank the IBRO team, especially Carolina and Adriatic, for their support for these webinars. And I also want to mention to you, IBRO ECC is having a call for papers for mini-series named Spotlight on Scientific Advances by Early Career Neuroscientists in the IBRO Neuroscience Reports. And we want to highlight the research by early career neuroscientists. We encourage graduate students and postdocs to partner with junior PI to write like review articles or submit like a research article to us. So if it is related to molecular and cellular neuroscience, you can submit to volume 1. And it is edited by myself, I'm Jacques, Lynn, and Manuela. And if it is related to systems and computational neuroscience, you can submit to volume 2, which is edited by our IBRO ECC chair Shrikant and Olamide. So and we will send you a link to fill in a feedback form. And after you submit the form, you will receive an e-certificate of attendance. And we want to thank you for your attendance today. And lastly, I would like to invite our IBRO's ECC chair, Shrikant, to say a few words before we end the webinars.
So first of all, many thanks to the speakers, so Simona and Thomas for their excellent talks, and to all the participants. I think this webinar was a resounding success. We had more than 300 participants. I think that's phenomenal, and you all learned a lot. There were a few general questions about joining IBRO and contributing to IBRO. So I'd like to provide some very quick pointers. So of course there are many ways to get involved with IBRO. So the most straightforward way is to apply for IBRO grants and fellowships. So IBRO provides many travel grants, training grants, short stay grants to visit labs as students. So that there's a host of financial support mechanisms that you should all really tap into. And another way to contribute is to attend IBRO schools. So IBRO organizes neuroscience schools worldwide, which focus on specialized topics like computational neuroscience or systems neuroscience or many other emerging topics. And of course, participating in IBRO workshops and conferences like the IBRO World Congress is another way to get involved. So the IBRO website really is a treasure trove of information. So I'd invite all of you to take a look. And feel free to also reach out to any of us for any questions. We are always happy to help. And of course, many thanks to Carolina and Adriatic and Angeliki from the IBRO team for all the logistical help and support. So yeah, I guess that brings us to the end of this webinar.
Thank you very much, everyone. Have a good day or good night.
Thank you.
Bye-bye.
Thanks. Bye. Bye.
Bye.